Hello, I'm Stephen Chernisky, Chief Science Officer for Unigen. Today we're going to be exploring a part of the EcoNet which is critically important for me and also important for you. We're talking about Unigen, which is the research division of EcoNet, and with me today is Dr. Edward Cannon. Dr. Cannon has been with Unigen now for about 2 years, and that's not exactly where you started, is it?
No, Stephen, it isn't. Started as a scientist, trained at Harvard as a molecular biologist, happened to be at the right place at the right time, Boston, when biotechnology took off, and was fortunate enough to be involved in founding and succeeding in profitable outcomes with 6 biotechnology companies in the Boston area.
What I'd like to talk about now is the differentiating factor between Unigen and Unigen and the conventional nutrition product companies that are out there. So could you tell us a little bit more about your discovery process and how an ingredient goes from the plant to the finished product?
Sure. So what we do at Unigen is we take what is essentially a pharmaceutical discovery approach. So when you go through our labs today, you'll see the same equipment that's used in every pharmaceutical research program. You'll see that we access the very same biology that gets accessed in drug discovery and development. But we use that technology in a very different way. We apply that technology to a proprietary collection that Unigen has of natural products that are derived from medicinal plants that have a long legacy of safe human use in the areas in which those medicinal plants are grown, as opposed to the pharmaceutical industry, which of course has very large collections of proprietary, non-natural, chemically synthesized molecules that they apply that very same technology, that discovery technology to. Those non-natural molecules, of course, have never seen the human body, and hence the side effects that limit their use.
At the same time, I want to differentiate Unigen and Unigen from what I would call the conventional nutritional product companies out there who don't have laboratories and who don't do research, and by the way, who don't have any proprietary ownership of the compounds that they're selling. Tell us a little bit about the patent portfolio.
Sure. Well, as I indicated, we use a pharmaceutical quality discovery approach. We are able to discover and identify and characterize molecularly the active components of these medicinal plants. So this is the molecular material that provides the health benefit. Because we identify the molecular component, we're able to actually patent that. So whereas others might have happened upon that plant in the past, no one prior to Unigen has actually discovered the molecular component responsible for the benefit. We also do extensive safety testing, and we follow that up with extensive human clinical trials. These, again, are pharmaceutical quality human clinical trials, randomized, placebo-controlled, double-blind clinical trials to demonstrate that, in fact, the benefit we hope to provide is indeed provided to humans.
Beautiful. Dr. Cannon, thank you so much for joining us.
You bet. Thank you, Stephen.
We're now in the Unigen plant library. This is a very special place and a place that's near and dear to your heart. This is Dr. Chi Jia, who is the CSO of Unigen. As Chief Science Officer, he oversees the collection process. In other words, he tells people what he's looking for, and then we engage a number of collection agencies to go out and find medicinal plants for you and your team to explore.
Yes.
You want to explain to us, how does that happen? How do you know what to look for?
Yeah, we started the medicinal plant collection back to 1997. We contracted with more than 12 companies and universities to collect medicinal plants from all around the world. We look into the specific medicinal usage of plants and to look into the potential functions and study them through our research program.
And this is an important consideration because a lot of times people will talk about natural products and they'll say, why are you guys doing all that analytical science? Why don't you just go with the traditional use that the plant has been used for?
One is novelty, and certainly there are so many active components in there, and looking into learning the usage does not yield novel components, such as protecting the functions of those ingredients was never being explored, and based on the traditional usage. so we can find the novel and the ingredients.
Beautiful.
Yeah.
Beautiful. So in addition to the traditional use that it's been used for, what you're saying is that your research team is able to identify other potential benefits
Exactly.
to be derived from the plant.
Yeah.
So I get it now. It's the combination of the traditional use and that information and knowledge combined with your team's Western analytical science to basically look at the plant, identify all the compounds and exactly what those compounds do. So about how many plants are in this library right now?
Currently, in this library, we have a close to 6,000 medicinal plants. And,
In this library?
In this library.
And
And we also have about similar numbers of plants collected in Unigen Korean site.
So altogether, close to 12,000 plants.
It has been collected,
And
yes.
then when you divide those plants into fractions, about how many fractions do we have?
We produced close to 14,000 extracts and more than 300,000 of fractions.
More than 300,000 fractions. All right. So this is, to your knowledge, is this the largest functional medicinal plant library that you know of?
I can say it's one of them because
Yes.
many of the universities and in different countries such as United States and Japan, Germany, they also have a large amount of plant collections.
Yes.
And but we certainly in the nutraceutical industry, we are the leading companies.
All right, so now we're in the second stage of the phytologics process, and I'm here with Mei Hong, and Mei has been collecting plants and cataloging plants and studying plants now for going on 12 years. Your job then is to take plant samples and to make sure that they are identified and a voucher specimen is created. Now, this voucher specimen is what would be in our library with the taxonomy, that is the identification of the plant in terms of genus, species, cultivar, and variety, and then all of that information goes into the library, and that information is shared with other plant libraries around the world. You've been doing this, and we're now adding about 1,000 plants a year. How do you manage all of that information?
Well, we have to, have a database to keep all the information come in. And first, when a plant come in, you have to check, match all the information, the name, the usage or the location where they collected, and put all the information in a database. At the initial stage, we just collect a certain amount, and until if you find something interest, we might go out for more, or maybe like we have a vertical integration or agronet, so we might grow our own.
Well, that's an important point, that when we do find a plant of interest, Bill Lee has purchased tracts of land in virtually all climate zones. So it looks to me like we're capable of growing just about anything that you may find that has medicinal activity that you want to pursue. In this laboratory, what we're looking at is the next step in solving the mystery. So we've already gone through the extraction process. We've already got that material then in an aqueous and an alcohol solution, and then we've got that dried into a powder, and then we've got that, and now we're getting down to the real nitty-gritty. Kat, tell us about this equipment.
This is our HTP, high throughput purification system. We take that organic extract or that aqueous extract and we run it through this, and what comes out are 88 fractions.
All right. So it's 88 fractions from a single sample.
Extract, yes.
Got it.
And so that's just, like you said, further isolating out the different compounds.
Now, LCMS for your stable compounds, GCMS for your
More volatile,
more
less
volatile
stable
compounds. Now, are there other natural product companies using this kind of technology in their discovery process? Not really. This is advanced technology which has allowed us to add about 1,000 plants a year to our plant library. This is the high throughput, this is the rapid identification, and it really does give us a tremendous advantage over laboratories that don't have this kind of technology. Now we've got to start looking at the isolation of individual compounds within that material. And what is this? Describe HPLC.
HPLC, after it goes through HTP, which simply further fractionates, separates out those compounds from the extract, it is tested in discovery screening. From those test results, we are told what are the most active fractions out of that. So we pull the most active fractions from that HTP plate, and we run it through HPLC.
Wonderful. And show us the information that you get from this, because this is, to me, the place where we're actually getting the first real clue as to what is doing what this plant has traditionally been used for. Kat, these peaks represent what?
Potential active compounds.
So potential active compounds, and there may be, what, 20 or 30 potential active compounds in a single plant. And so that means that we need to follow this mystery a few more steps before we get our answer. So you can imagine in this, as the mystery is being solved, you can imagine with the information coming out of HPLC, we've got a bit of a traffic control issue here. Dr. Jifu Zhao, who is a professor of chemistry in China for over a decade, is actually helping to manage the traffic control of information from HPLC to the other technologies like GCMS and LCMS. Now, Dr. Peng Zhao is the master of this arena. So, Dr. Zhao, is it accurate to say that in the bio lab, we're basically finding out the composition of a plant, and with your equipment here with proton NMR, you're actually able to identify the molecular structure of those compounds. Is that right?
Yes. Basically, every molecule has the composition with carbon, proton, oxygen, nitrogen atoms. This, with all the color data you collect by this machine, you actually can kind of visualize the detailed structure.
So a three-dimensional picture, if you will, not just the composition, but the molecular structure in 3D, which is really got to be awfully exciting. I mean,
it's very important
and precise
for research.
Okay. Well, you know, I've had the chance to tour a lot of natural product laboratories all over North America, and even some in Europe. And I've seen some pretty good laboratories with some good biological and biochemical capabilities, but I've never seen another lab anywhere with proton NMR. Possibly that's because this is new technology, and I'm also thinking that it's possible that we care more and that we're looking for true discovery, not just imitating other products and not just kind of quality control, but actual discovery at the molecular level. So thank you for all the hard work that you're doing, and let's keep going. Here we are in discovery and screening. I'm talking here with Lydia Brownell, and we're looking at a chip scanner. Now, this chip scanner, you load a 96 well plate, which gives us, what, 96 experiments, and you can load multiple plates in this automated scanner. And what does this tell you?
The plates we use because they're very versatile. We can do, the IC50 concentration. We can, do our curves out of here. We can get a lot of information. When we load them on the instrument will do the reading at specific wavelengths based on the program, and then we can walk away, come back later, use the software, do the calculations, and extract our data.
Now, interestingly, then it gets basically handed to you because Lydia is involved in designing the clinical trial. So tell us about that.
Normally, we start with small pilot trials that we run through universities and CROs and here in the U.S. and some of them, and we run in Korea and other places. Once we get the results from those trials, then we develop bigger trials. Maybe we look at more markers, different populations, and that's basically at the last step.
So, in other words, if you're a Univer associate and you're looking at a product that actually ended up in your hands, you've got to have an incredible sense of confidence of where that product came from. Because it wasn't just something which someone thought, oh, they've used this herb for that purpose, and let's put some of that in, and let's put some of this in. These are way beyond what I would call conventional nutrition, and we're now into advanced cutting-edge science that produces products that have efficacy that's been proven at the biological, biochemical, genomic, and now clinical trial level. And that's really very, very unique. Well, as you can imagine, discovery and screening is a laborious process. I mean, I'm here with Brandon Corneliuson, and you're working in discovery and screening, and you've got a lot of work to do. I mean, if you look at Protectin, which is one of our best science to date, in order to find those two plants that had that profound effect on joint comfort, mobility, and range of motion, you had to screen something like 1,250 plants, and each one of those plants had to have multiple assays.
So this is an automated liquid handler. It can actually do 96 wells at a time.
All right.
So it can speed up the process significantly, 5 to 10 times faster.
You want to demonstrate this for us?
Yeah, sure. Within each 96 well plates, there are many plant extracts, and what the automated liquid handler will do is will pick up different reagents, and it will dispense them into each of the 96 well plates to find if there's any sort of reaction that can then be further utilized down the road with any specific target we're looking at.
Okay, finally we're in the genomics lab, and genomics, as most people know, is the study of the human genome. In this case specifically, the effect that plant compounds have at the DNA level. So I'm here with Dr. Mesfin Imam, and Dr. Imam, could you tell us exactly what you're doing here?
The genomics lab is part of the discovery and screening department. Any procedure that we conduct in this lab will help us understand the mechanism of action, new application, or if there is any potential side effects of our novel ingredients. And to do that, studies are done on specific genes. Okay? Those genes have they have a great or link association between immune functions, metabolic process, inflammation processes. So by doing that, we should be able to determine, that a specific bioactive component in question either up or down regulate the expression of those specific genes that we want to see.
Now, I assume this is something that you would not find in any conventional nutritional product company. So this is really highly advanced technology, which again, I want to say, sets Univera and Unigen apart from your normal conventional product company just taking vitamins and minerals and putting them in a bottle and selling them. So now we're in quality control, and Michael Zahn is the master of this domain. What we're going to be talking about for the next couple of minutes is what quality control really means. Who sets the standards for what the exact precise ingredient ought to look like?
Recently, the FDA has come out with some GMP guidelines that dietary supplement companies are going to have to adhere to, so we are strongly following those.
You guys keep this lab in peak condition, regular calibrations according to FDA guidelines, is that right?
Correct.
So this is something which you have a whole team here dedicated to making sure that every ingredient is as pure and as potent as we say it is.
Absolutely.
If I was to summarize this entire lab tour in 3 words, it would be discover, develop, and deliver. The EcoNet family of companies, especially with Unigen as the discovery channel, is looking through the planet for medicinal plants that have been used but have been poorly studied. So we might find plants that have been used for a medicinal purpose for maybe 1,000 years, but they've never been rigorously studied to identify their safety and efficacy. If you understand the commitment to quality, and you've seen the quality control and equipment here, you've seen the dedication to identifying the specific active ingredients and maintaining those active components at a specific level. That's called a standardized extract. All of this is available from Unigen through Univera to you.